So this ought to be pretty easy. Everybody ought to see this one. Here comes the confusing one. This shows you, now look at this, take it step by step. Here's the blue and the yellow, right? The blue is exon 1 and exon 2, and the yellow is the intron. Here's the U2 RNA that's recognizing the branch point A. There's the complementarity between the branch point A and the primary transcript and the U2 RNA. It's lined up, right? Now U1 is gone. It's not in the picture here, but you notice there's U6. Remember the ball here, after everything happens down here, U6 is still there. So it's part of that nerve. Lotus, they show you actually U6 is acting with U2 there a little bit. Well, that's what happens. So here's U6. It has complementarity to a region in the 5' end, right? Remember now, there's 5, we'll recognize this. This is the consensus thing. There's that conserved G all the time. It's on 5' end of the intron. Who recognized that first? U1 SNRP, right? But then remember, the U1 SNRP got blown out when that triplet came in. And one of the proteins in that triplet SNRP was U6. And it recognizes some of the same stuff. It doesn't have as much complementarity. It can bind a little bit more loosely. It doesn't have the big complementarity that U1 has to recognize it, but it does have some. So U1 comes in and recognizes it. It's then displaced by this guy. This guy, U6, has complementary to this. It also has complementary to the RNA that's in U2. This sequence here, they don't tell you, but that's the U2 RNA sequence, right? This is the U1 RNA sequence up here. Notice all the U's, by the way, why they're called uRNAs. So here, this U2 has complementarity to part of the RNA in U6. U6 has complementarity to part of the 5' end of the intron. U2 also has complementarity to the branch point A. And when you fix all these complementarities together, right, when you put them all together, look what it does. It takes that A, branch point A, and puts it right there, sort of lines them up. So the biochemistry that happens, the closest bonds, right, are that hydroxyl, 2' hydroxyl, and the phosphate, the phosphoester that's right in there. They're just right there next to each other, and the biochemistry just happens because they're pushed. This hydrolytic attack to that hydroxyl group on that phosphate, phosphoester bond. So all of this stuff, and all of this movement, and all this complexity, and all of this evolution that's so complex, works just to line those two up. And the other thing is, remember I told you how that A popped out? Well, look, if you look, there's complementarity between all these nucleotides, but the A that sits in there, there's no complementarity guy here. So when this complementarity forms between the U2 RNA and that branch point A, it kind of takes that A that doesn't have a complement, doesn't have a partner, it kind of pushes it out, just like a belly button. So that hydroxyl group is sitting there right at that phosphoester, ready to be cleaved. Okay. Now what happens is, look, the cleavage has happened, right? There's that branch point A. That's the branch point A. The layer is formed. And now what has to happen is this guy here has to be put next to that guy there. How does it happen? Well, I'm not going to go through all of this, but you can see there's actually a movement in there. There's the U6 is now, still has complementarity there, but now there's another guy that has complementarity. U5, actually, his role is to keep, once you cleave off this exon 1, right? His role is don't let that thing go. It's got to stay in there till it ligates. And also, get it around near that guy there. So notice you see this precursor now is kind of whipped around this way. So we have this new complementarity. This complementarity is exchanged. There's actually another one in there now that they're not showing. What they're just showing is you have to line these two up. You put this free hydroxyl very close to that phosphoester, hydrolytic attack, cleaves it, transesterification, and you have these two ligated, exon 1, exon 2 ligated. Now, I don't expect you to draw all of this. I won't ask you that in an exam. I wouldn't want to have to grade it. It'd be terrible. So, but I want you to understand how intricate that movement is. And, you know, this is like one of the dances of biology. It's pretty fancy stuff. Okay. What else do I want to tell you about that? Just thought of something I forgot. Oh, remember over here I said, just in this little cartoon here, I said, ATP, 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 ATP. What's all that ATP for? What's all that ATP for? Why do you need it? You don't need it to make this ester. told you the transesterification doesn't require it. What's it for? Well, remember, all of this interaction here, right, all of this RNA, in order to move, in order to get rid of one and get this guy, in order to get this tail flipped around and everything, what you have to do is you have to have energy to break that RNA bonds. Right? It's like a helicase, like an RNA helicase, not quite, but it is a helicase. And it breaks those bonds to pull that RNA away. And so when you look at this picture way back here that we showed, and here after everything's done, right, we already made this, now we have still ATP. What's happening? Well, remember U2 was interacting with U6, we got to pull them apart so they're ready to go. They have to be separated, right? How do we get them apart? We need ATP to pull them apart. So all of the ATP is used to get this machinery and this ATP complementarity that lines up everything for the spliceosome to work, and then when it's done, you have to reprocess them so you can use those again. There's only 100 million, right? You have to reuse them over and over again. RNA processing. Does anybody have a question? Spliceosomes. I'm not going to ask you to draw that picture. Like I said, I won't ask you to draw this. But I might ask you a question about what it's all about. You know, why is there this complementarity between those small nuclear RNAs? What's the function? And you have to at least know that the function is to take and jerk around that HN RNA or that primary messenger transcript so that it lines up the proper nucleotides for the cleavages and the ligations. Right? You have to at least understand that concept. Okay. This just goes to show more. This is the same sort of stuff, and this just shows a little more how these changes, right? You can see that these two blue regions in this step where the VPP is there are far apart, and then when the VPP comes off and you get this other U2 RNA coming on, look, now those are in hybrid. These are not in hybrid, these are in hybrid. These are not in hybrid, these are in hybrid. So there's all of this movement, and that's what causes this thing to move around and slide around the line. Okay. So, removal of intervening sequences. We talked about splicing, we get these things out. Well, it turns out there's a lot that this splicing is really important. If you mess it up, if it gets screwed up, right, it can cause problems, it can cause diseases. And this is just one example. You notice this guy here, three exons and 2 introns, that's the globin gene. And that's about right. These guys are about 100 or 200 nucleotides each, one bigger intron in there. And normally what happens is they're cut out, and the yellow region, right, so the cleavage goes from where the red is, here, 5' end, 3' end of the intron, this guy's cut out, same here, exons put together. There are a group of diseases that are known as beta thalassemia, and they occur in humans, and they involve mutations that mess up this processing event there. There are several of them. I'm only going to show you about a couple of them here. One of them is this guy here. So what happens is this particular thalassemia here is there's a mutation in the 3' recognition site of the intron. So remember, we have the 3 sites to recognize it. The one here is lost. So the spliceosome begins to set up, and U1 and U2 recognize this in the branch point, right? And they begin to splice. And since there's not a 3' recognition site in order to religate these two, what happens is the ligation goes to the next 3' site, which is here. So what happens is when you cut out, in this mutation, rather than just cutting out each of the introns, right, it cuts out this whole region here, which is one of the exons. So you generate a messenger RNA that's missing an exon. Well, the beta thalassemias are sort of interesting diseases because they're generally not fatal, most of them. I guess there could be a fatal one, but we wouldn't know about it very much, right? Because if you inherited a fatal mutation, you probably died before you were born. I guess you could have fetal globin, but anyway, you die shortly after you were born. And we might not know. But anyway, these are interesting because this protein that only has this encoded by exon 1 and exon 3 of this globin is enough to carry oxygen, right? It does carry oxygen around. The problem is, because it only has those 2 introns, the Km for the binding of oxygen to that particular globin molecule is much higher. So it doesn't bind oxygen sufficiently. So when this globin moves into red blood cells to different cells, in your body is not carrying enough oxygen. So this guy with beta thalassemia is, short of oxygen, short of breath. But it's a genetic mutation. It's a mutation that occurs in that 3' recognition site in chromosome. And this occurs. This is real. Somebody didn't make this up. There's another beta thalassemia that is here. And what happens is it's sort of like a gain of function mutation. There's a mutation within this intron here that generates a 3' recognition site. Right? An intron 3' recognition site right there. So now when this intron processes out correctly, this one, so that's correct. This one begins at the 5' site, but rather than going to the 3' side of the intron, it goes to this new mutated site that has this sort of a gain of function 3' intron recognition site. So what happens is that since it ends there, it cuts out this there, and it makes a new globin that has an extra piece. This piece here just happens not to have a stop sequence. It happens to be triplets in sequence, so that the gene that comes out, or the RNA that comes out, has intron 1, intron 2, intron 3, plus this extra, but it codes for a protein. This protein here actually binds globin, right? It binds it better than this globin molecule here. It has a lower Km and it binds it better. The problem is that when it gets to the tissues, it doesn't want to let it go. So your globin has lots of oxygen in your blood cells and it's bathing your tissues, but it doesn't want to let that oxygen go, so your tissues are still starving for oxygen. Either way. So both of these result in low oxygen delivery to tissues in the problem. Okay, I'm not going to do this because that just goes on beyond that and says, hey, you can put one mutation here and then have another mutation for start, and then you have essentially 4 exons. All of these exist. They're all beta thalassemias, and as doctors, you'll diagnose that probably sometime in your life. Okay, so if you don't take these out properly, it's a problem. Okay, so here's a lot of